thanks guys for coming today. My name is Krista and I'm with the Alberta Plant Health Lab. So we're at a crop diversification center north in Edmonton, Alberta. So today I'm just going to kind of go through that, like who are we, what are we doing, why are we developing a new lab. So Alberta Agriculture identified the need for a diagnostics lab for the agricultural industry. So we had somewhere for people to send in samples when they see kind of strange things in their crops or you're not sure what you're dealing with. So we've been working with that. We started up the lab. It's been about 2 years in the works, and finally this year, about a month ago, we started accepting samples. So we're out here with you guys to make sure we also reach southern Alberta. So we're located out of Edmonton, but we're accepting samples from the entire province, so anyone can, through the right channels, which I'll tell you about, send in samples to us. So the services we offer are mainly for diseases, weeds, and insects. So weed identification, insect identification, and then what I mostly brought for you today is pathogens to look at. So we have some samples at the end and a few questions for you guys. So the samples we accept are plants for diseases or for weeds, the insect samples, and also some soil, just depending on what we're looking at. As part of the government, we're not looking to function in competition with private or commercial seed labs, so we don't duplicate the services they offer. So we don't have a chem lab, we're not going to be doing fertility or salinity testing, as well as some of those routine tests. So for example, Clubroot, which is what I brought for you guys to see today, is you don't have enough of it down here, so I brought it with me from Edmonton. Routine Clubroot testing for kind of those common pathotypes that we see around central Alberta. We won't be doing those because that's one of our commercial lab's biggest businesses. But for example, if one of you guys was growing a Clubroot resistant crop and you were still noticing a lot of galls or Clubroot symptoms in your crop, that would be an ideal sample to send into the Alberta Plant Health Lab. From there, other things that we exclude are the seed testing services. So we don't deal with seeds, so purity or vigor. And right now we don't have a chem lab, so we can't do herbicide testing right now. But in the future, we're hoping to expand to that. So for the samples we do accept, the way to get them to us is through, we have a few groups. So we're a small lab right now. We have 3 permanent staff and 3 seasonal staff, so a total of 6 of us in a building right now. We're accepting samples through your ag fieldman, so if you know your county fieldman well or you want to get to know them so you can send in samples through them. Applied Research Association, so that's how Farming Smarter actually found us. If you see something funny, you can talk to Ken or someone here about getting a sample out to us. Or Agriculture and Forestry, so anyone from the Brooks area, or if you're familiar with CDC South, they can also get samples over to us. So that's kind of what we're looking at. For diagnostics, what we mainly run is we start with kind of like whole plants and field level. So often we get pictures sent in or whole plants. So what we kind of start with is something like this pea plant where, oh, thank you. I'm not used to the wind here yet, guys. I'm from Edmonton. This is still new. There, my plants can hold it down. So what we see is something like this, where we get a whole plant sample sent to us, and so we immediately start looking for what's not right about it and what's going on. And so we'll start with that, and then often microscopy is the next step. So if someone sent us, say, a canola sample that had a lesion on it and they weren't quite sure what it was, put it under the microscope, and if you start to see those little black dots, that leads you towards knowing that you might have blackleg. So I brought some plates of blackleg you guys can feel free to look at. And what you'll notice is the next thing we might do is culturing, so growing them out, and they'll start to even produce the little black pycnidia of blackleg on a plate. They don't even need a leaf, they're happy to start it. Or for example, sclerotinia in canola. I know one of the other stations here also has sclerotinia to look at, but they'll grow those little sclerotia that you see that show up in your stems. They'll grow them right on the plate. They're more than willing. So that also helps us start to figure out what's wrong with the crops. And then from there, we also go into some more molecular techniques. So we're doing a lot of work with DNA lately. And one of our big ones is PCR, so polymerase chain reaction. So that one amplifies some of your pathogen DNA, so you can kind of get a positive or a negative test result. So for example, if someone sent us soil and clubbed roots from canola, you can use PCR to know if you have it or not. And then qPCR is the quantitative version. You can tell how many spores are in your soil or in your gall. So those are the kind of tests that we've been running a lot of. We also work with Pest Surveillance. So for example, down south, I know the Fusarium head blight survey is pretty big. So we got some from corn here. So the big kind of tick off for us is if you see this pinkish red color, it's fusarium. So these are from southern Alberta corn samples from last year's survey. So we've been growing them out, and yeah, a lot of what we see is different fusarium species. And then we can use that molecular sequencing to find out, oh, it's starting to rain, but use the molecular sequencing to determine exactly which fusarium we have, if it's graminearum or a different one. Or is anyone here familiar with Jimsonweed? That weed they found last year in a lot of canola crops, mostly in my area, coincidentally of course as well. Well, what it is it's a big poisonous plant that came in with some canola seeds. So they noticed the normal canola crop and then this big towering plant that they call devil's trumpet commonly, because all parts of the plant are poisonous. It produces like a big spiny pod full of seeds. And if you get that mixed in, it's really hard to separate it out of your canola. And then we had some people start calling in about ornamentals in their yard that look really similar and being concerned if this was the same plant. So through molecular testing, we could figure out down to one DNA base pair if it was the same or if it was different. So we had a lot of samples come in that were that, but also some that were just very similar. So that's kind of the benefit of that. Finally, about sending your samples in. When you want to submit a sample to any of these guys, just give them a little bit of notice, but the best thing to remember is kind of your packing and keeping things fresh. So if you want to send me a plant sample that has something going on with the roots and it's a fresh plant, you'll want to maybe wrap the root in some paper towel to keep it damp and send it in a plastic bag. Or if you want to send me soil, the opposite. I want dry soil. So you don't want to get that kind of mold and stuff growing after the fact, because when I plate it out, you'll just end up with a lot of mold happening. Those are kind of the big ones for us, and what we're working on next year is also more with Clubroot, because there's so much in my area, is that 5X, so the new pathotype. We're doing a little more monitoring for those fields around the province, and our lab has developed a molecular technique to test for if you have the more common types of Clubroot, or if you have those more virulent strains that we don't have a resistant cultivar to yet. So does anyone think they know what this is? You can come closer if you want. You can hide under the tent even.
Oh, why don't we move the table back?
Ooh, yeah, I was gonna say, let's just move the table back. Yeah, let's. Yeah, there we go. That's my mind. There. People can come inside now and hang out.
Would this be the new Phenomyzer root rot?
Yes. So what you'll notice is that characteristic kind of honey caramel color. That's what tells you that you're most likely looking at a Phenomyces. So
The reason I know we got it.
oh, well, there we go. So an expert, perfect. Yeah, so this is the kind of thing we're looking at, or a Phenomyces or a Phenomyces and Pythium kind of in a complex together. So we're working on that. So we also do a little bit of research. So Saskatchewan and Manitoba both also have labs, and our lab also does some of the research through Alberta Ag still. So our research just feeds back into diagnostics. So we work on new diagnostic techniques or developing primers for these pathogens, so ways to be able to differentiate if you have Pythium versus a Phenomyces or if you have both. Yeah.
Could you take a soil sample and find out if we got that in our soil? Let's say do you want to see before you want to see peas or lentils?
Yes, I believe there is soil sampling because it is a soil disease, Phenomyces.
Yes,
so you should be able to soil sample and test for that one. Oh, now everyone wants to come into my tent. Got this. Yeah, and you guys, also to note on my little clubroot for you guys is you can see the little club roots growing in the soil if you want. And we can't dig them out just because we don't want to move the soil around. But I brought some in jars you guys can pass around if you want. This one's my favorite. So, yeah, they're preserved in ethanol. They're pretty good. They're pretty clean, but just in case. Yeah, so those are cut from plants that were, these are all sown at the same time in soil. This is Westar, so a really old variety of canola because it's very susceptible. It doesn't have any inherent resistance to clubroot, so we grow that in our clubroot experiments a lot. So those are galls off the same batch of plants. And what you'll notice is this one probably has the biggest gall, and it's the smallest little plant happening right now compared to someone like these ones. It's pretty stunted by disease, and then I clipped these roots off of the ones that were in the same box. So that gives you an idea of what's under the soil, even though we can't dig them out right now. And then I just have a few other examples of samples we get. So often, this also goes with sample packing, of trying not to leave your samples in the truck for too long because they dry out like these ones did when they traveled here with me. And for example, sometimes we see leaf streaking, so we test them for things like wheat streak mosaic virus and to find out what's there. These ones came back negative for it, and when we actually plated them out, some of them had fusarium is what was causing it. So these ones are fusarium. A fusarium from corn to look at. Brought some Verticillium dahliae from potato fields in southern Alberta, so it's a fairly common pathogen in soil in potatoes. It's a little different than the Verticillium in canola that people are talking about right now. That one's Verticillium longisporum, and so that one's a hybrid pathogen versus this one. So you guys can pass these around if you want. Just shows you also the different cultures that we see in the lab when we start growing things out. And then I have the blackleg in canola if anyone wants to look at it. Yeah, and you guys can feel free to take a sheet too, and it has all the information about submitting samples and how they get to us, what we accept, and what we're working on.
Now we need some positive news.
Some positive news?
Yeah. We just seen problems.
Well, I don't have the most positive 10, do I?
Yeah, it's raining.
Is that we're working on diagnostics to try to make these things better or to try to detect them. We're also working a little bit with some in-field diagnostic techniques. So for Clubroot, for example, we're trying to get something developed that in the field you could detect whether or not you have Clubroot without taking a sample back to the lab. So yeah. Thank you guys. If you have any questions, let me know.